I'm Laura Rance, Vice President of Content for Glacier Farm Media. I'm joined here today by Christina Eink. She's a plant breeder with Agriculture and Agri-Food Canada based in Saskatoon. Christina is here to tell us about her work developing new oilseed crops for prairie farmers.
Thank you, Laura, for the introduction, and hello, everybody. Today, I will talk about some of our work on breeding of the oilseeds Camelina and Carinata at the Saskatoon Research and Development Center. In my presentation, I will first talk about Camelina and our work on increasing its seed size, and I will introduce you to winter Camelina. In the second part of this presentation, I will give an introduction to Carinata, and I will talk about our breeding program and the development of ultra-high erucic acid Carinata, Carinata resistant to Clubroot, and Carinata hybrids. The last bit of my talk will be about work that my colleague Isabel Parkin and I are doing on the development of a molecular breeding platform for Carinata using a nested association mapping or NAM population. For a number of years now, Camelina has been considered as an alternative oilseed crop for the prairies. Camelina originates from the steppe regions of southeastern Europe and southwestern Asia. It is an old crop that has been grown in Europe for centuries before it fell into oblivion, but it is now regaining interest as an alternative oilseed crop. And this is in part due to its agronomic attributes such as a short growing cycle, frost and drought tolerance, which give it an excellent yield potential in short growing season environments and on lighter soils. Camelina is also resistant to a number of Brassica pests and diseases such as blackleg and flea beetles. And the fatty acid profile of Camelina oil is quite unique and makes the oil suitable for a wide range of applications from human and animal nutrition all the way to industrial applications. Even though Camelina looks quite different, it is a member of the mustard family. This table compares Camelina with canola and flax in terms of seed oil and protein content and the percentage of the major fatty acids in the seed oil. Considering that Camelina has not been extensively bred compared to crops such as canola and flax, it already has a high seed oil content of around 41 to 42%, depending on the weather conditions during seed filling, and a high protein content of around 30%. Camelina oil has a high content of the omega-3 fatty acid, linolenic acid, which is not as high as that of flax oil, but much higher than that of canola oil, and which is unusual for a Brassica, Camelina oil also has a considerable amount of gondoic acid. The erucic acid content is generally below 3%, which is below the maximum level permitted in edible oils. Even though Camelina oil has a high percentage of unsaturated fatty acids, it is surprisingly resistant to turning rancid, which is due to its high content of tocopherols, also known as vitamin E, which act as antioxidants. Despite its agronomic potential, particularly for lower quality land, Camelina has never been grown at a large scale. And the main reason for this is that there was never a real market for the oil or the meal. And quite recently, that has changed drastically. While the initial focus of Camelina development was on industrial applications, such as biofuels, the realization of the unique properties of Camelina oil have led to a shift to food and feed applications. Current markets for the oil include salad oils, functional foods and nutraceuticals, personal care products and cosmetics, but particularly the animal feed sector, and here the aquaculture market, and an increasing market for Camelina oil as a supplement or feed ingredient for pets and companion animals, such as horses. The meal received CFIA approval for poultry, and research is underway for specialty dairy products. In other countries, Camelina meal is also approved for cattle and hogs. Camelina breeding in Western Canada started in 2007 in Saskatoon, and in 2013, the first Canadian Camelina cultivar was developed and released under the name Midas. The breeding objectives for Camelina are, for the most part, quite similar to those of other oilseed crops, with priorities given to a high seed yield and a high seed oil content. We also select for disease resistance with downy mildew, sclerotinia stem rot, and aster yellows being the most important diseases in Camelina. And we certainly select for lodging and shatter resistance. The main challenges in Camelina are its small seed size and the lack of post-emergence weed control options. Compared to cereal seeds and even canola seeds, Camelina seeds are very small. They are about 1/3 of the size of canola seed. And this small seed size makes seeding and combining, but also crushing of the grain for oil challenging. We are working on the development of larger seeded varieties that we hope will have an improved seedling emergence at higher seeding depths, improved combinability, as well as an improved efficiency of the crushing process. The last process or the last stage in a breeding program is the evaluation of advanced breeding lines in replicated yield trials. And this table shows the results from one of our replicated yield trials, in which we evaluated the agronomic performance and seed quality of some larger seeded breeding lines from our program in comparison to the commercial Czech Midas. All numbers shown in green are significantly better than the corresponding values in Midas. And what you can see here is that all lines presented in this table have significantly larger seed than Midas, ranging from 23% to almost 40% larger. And this increase in seed size has no negative effect on the oil content, which is often the case in Camelina. And while the days to maturity and the protein content are also unchanged compared to the Czech, a number of our advanced materials show seed yields that are considerably higher than that of Midas, with up to 22% higher yields here in Swift Current for entry 7. Data from other locations do look similar. And one reason why I wanted to show these data is also to demonstrate that there is still ample room of improvement of Camelina through breeding. Next to spring Camelina, we have recently expanded our program to include the development of winter Camelina. Winter Camelina is a fascinating crop for several reasons. For one, it is extremely winter hardy. It is just as winter hardy as fall rye. It also has a unique growth pattern at the rosette stage, with leaves lying more flat to the ground, which make it more competitive with weeds compared to spring Camelina. The early development of winter Camelina also means that it avoids a lot of the diseases we see in spring Camelina. The oil content is higher, and the oil contains a higher percentage of alpha-linolenic acid, which makes the oil of winter Camelina even healthier than that of spring Camelina. Growing winter Camelina also comes with a wealth of environmental benefits. It is a crop that covers the ground over the winter, which contributes to a prevention of soil erosion and an uptake of excess nitrogen. And because winter Camelina is one of the first flowers to bloom in the spring, it is also one of the first feed sources for pollinators. Winter Camelina is one of the, if not the only, winter oilseed that can successfully be grown on the Canadian prairies, and with that has the potential to facilitate a true winter crop rotation when grown in rotation with cereals. I would now like to switch gears and talk about Carinata. Carinata is also known as Gomenter, Abyssinian, or Ethiopian mustard, and as its name suggests, its center of origin is Ethiopia. Carinata is a member of the mustard family and a close relative of canola, and it is derived from the interspecific hybridization between black mustard, Brassica nigra, and cabbage, Brassica oleracea. One of the most interesting characteristics of Carinata is that it is extremely tolerant to abiotic stress, especially heat and drought. Most Carinata germplasm is high in erucic acid, and this is why it was developed at the Saskatoon Research and Development Center by my predecessor, Kevin Falk, as an industrial feedstock crop for the production of biofuels, particularly biojet fuel, and as a feedstock for oleochemical applications. The biofuel produced from Carinata oil is certified as sustainable by the Renewable Energy Directive in Europe and the Roundtable on Sustainable Biomaterials. Over the past few years, Carinata has made the news internationally as a feedstock crop for the production of biojet fuel, and also as a means to help the aviation industry reduce its footprint. A Carinata breeding program was established in Saskatoon in the 90s with the goal to develop varieties that are adapted to the growing conditions on the Canadian prairies. One of the major breeding objectives is to develop varieties that mature in our short growing season. Current varieties mature about 7 to 10 days later than canola. Other breeding objectives include good agronomics, a high seed oil content, large seed, disease, and shatter resistance. In our program, we also have a number of projects on the development of germplasm with specific traits of interest for the industry, such as material with ultra-high erucic acid content, with low or high glucosinolate content, clubroot resistance, herbicide tolerance, and last but not least, Carinata hybrids. Much of our work takes advantage of the fact that Carinata is a member of the so-called triangle of U. The triangle of U describes the relationship at the genome level between the Brassica crop species canola, or Brassica napus, condiment mustard, or Brassica gensea, and Brassica carinata, here shown on the sides of the triangle. And all three species were formed through the hybridization of two ancestor species, which are shown at the tips of the triangle. What this triangle shows is that Carinata shares one ancestor with both canola and condiment mustard, and that means it also shares a genome with them. These shared genetics allow us to integrate traits from the other two more established crops into Carinata through so-called interspecific hybridization. And it also means that any knowledge we gain in Carinata can potentially be transferred to the other Brassica crops. The transfer of traits through interspecific hybridization generally takes a long time compared to genetic modification, but the resulting plants are in many jurisdictions not regulated, which is a clear advantage. In a project that is of relevance in the context of providing renewable alternatives to non-renewable resources, we developed Carinata with ultra-high erucic acid content. Erucic acid is an interesting feedstock for the oleochemical industry with more than 1,000 potential patented applications. Because the pathway for the synthesis of erucic acid, which is shown on the right-hand side of this slide, in brassicas is well characterized, we had a good idea of the genes that needed to be introgressed into Carinata. We first transferred the gene from Brassica gensea, or condiment mustard, and after that a gene from Brassica rapa, or Polish canola, and ended up with a stable Carinata line with erucic acid levels of over 58%, from 48% in our starting material, and compared to just over 40% in current commercial Carinata varieties. This is the highest erucic acid level reported for Carinata. As a member of the mustard family, Carinata shares many of the same diseases and pests with other brassica crops like canola. And while Carinata has good resistance to blackleg disease, it is highly susceptible to clubroot. And clubroot, as you all know, is one of the most serious diseases of brassica crops worldwide and has become a major threat to the production of canola in Western Canada. In a project that had the goal to identify clubroot resistance genes in Brassica nigra, which is one of the progenitor species of Carinata, two genes were identified that confer complete resistance to a broad spectrum of clubroot pathogens, races. In collaboration with the team of Gary Pang, we have successfully transferred this clubroot resistance from Brassica nigra into Carinata. And the pictures in the slide show our clubroot resistant material on the right with very healthy roots after inoculation with the clubroot pathogen, and the clubroot susceptible check canola variety Wester on the left showing severe clubroot symptoms. Another project in our program that makes use of interspecific hybridization is the development of Carinata hybrids. Hybrids are simply the offspring of two unique true breeding parental lines. They display heterosis or hybrid vigor, which makes them more productive or higher yielding than both parents. A hybrid system is composed of three components. First, the seed parent, or the A line, which has been bred to be male sterile, which means it cannot pollinate effectively. The maintainer is genetically identical to the seed parent, but male fertile and is used to produce seed of the A line. The third component is the restorer, which is able to restore fertility when crossed to the seed parent, which results in fertile F1 hybrid seed, which is then sold to the producer. At the Saskatoon Research and Development Center, a Carinata hybrid system was developed by transferring the widely used Ogura cytoplasmic male sterility system from condiment mustard. And while we are working on optimizing the restorer line, our industry collaborator Newseed has already been testing the first prototype hybrids. As you can see in this picture from a trial at Milestone in 2017, the first set of hybrids, here shown on the left side of the picture, showed noticeably greater vigor than the open pollinated lines on the right-hand side that were grown in the same trial, and also achieved considerable yield gains over the commercial check. Nearly all breeding programs of the major crop species use molecular markers to make the selection process more efficient. Currently, however, the application of molecular markers in Carinata breeding has been limited to a few traits only. In a collaboration with Isabel Parkin's lab, we have been working on the development of a large set of molecular markers and the corresponding assays in order to be able to identify favorable trait combinations in plants at an early stage in the breeding program. For this, a large mapping population was developed by crossing 50 genetically diverse lines with a common reference line and subsequent selfing for several generations, which resulted in a population of 2,500 individual genotypes. These were grown in 2017 and 18 at our research farm and phenotyped for a large number of in-field and post-harvest traits. For this, we used hands-on measurements, lab analysis, as well as remote sensing. Parallel to the phenotyping efforts, the 2,500 lines were genotyped, and the genotypic and phenotypic information were brought together in a so-called genome-wide association analysis, which identified variants in the DNA, known as single nucleotide polymorphisms, or SNPs, that were associated with most of the agronomic and quality traits that we looked at, here shown for base to flowering. These SNPs can readily be converted into molecular markers, which we plan to routinely use in our program in the future. The Carinata NAM population has proven interesting also from a pure practical breeding standpoint. What we did was we tested a number of NAM lines in replicated yield trials in 2018 and 19. And as the results in this table demonstrate, we identified unexpectedly promising germplasm, such as entry 6378, with significantly higher seed yield and oil content compared to the commercial checks A110 and A120, which are Carinata varieties that were developed at the Saskatoon Research and Development Center and licensed to industry. With this, I've come to the end of the overview of our program, and I would like to acknowledge the people that I work with. First of all, the people in my team, Isabel Parkin's lab and her team, our industry collaborators, Smart Earth, Camelina, and Newseed, as well as the funding that makes our work possible. And I would like to thank you for your attention, and I'd be happy to take any questions.
Christina Eink is a plant breeder with Agriculture and Agri-Food Canada. Thank you, Christina.